Okay, in this video we're going to talk about muscle fiber contraction. And muscle fibers contract in 4 coordinated steps. It starts with nerve stimulation that leads to the production of an action potential in the sarcolemma. This action potential or electrical impulse is propagated or spread along the sarcolemma and then ultimately causes a rise in intracellular calcium levels released by the sarcoplasmic reticulum. Now, steps 1 and 2, like nerve stimulation and action potential, these occur at something called the neuromuscular junction, which is the point of communication between nerves and muscle cells. Now, steps 3 and 4 are basically those things that link the electrical signal up to contraction. And then after step 4, we get something called excitation-contraction coupling, where 3 and 4, which is the excitation component, leads to the production of a contraction, which we'll talk about later in this video as well. So skeletal muscles are stimulated by somatic motor neurons. A somatic motor neuron is basically a nerve cell whose cell body is going to be in the brain or spinal cord, but it has a long thread-like extension called an axon that goes away from the brain and spinal cord or central nervous system out towards the skeletal muscle, whether it's in your face or neck or somewhere on your trunk or limbs. And these axons can divide into many branches as they enter the muscle to stimulate multiple muscle cells or fibers. Now, the axon branches end on a muscle fiber, and they form something called the neuromuscular junction or motor end plate. And each muscle fiber has at least one neuromuscular junction with one motor neuron. That way it's able to be stimulated by your nervous system. So the axon terminal is the end of the axon, or the end of that long filamentous extension of the somatic motor neuron. Now, the axon terminal is separated from the muscle cell by a very, very narrow space called the synaptic cleft. And within these axon terminals, we have basically synaptic vesicles that store a lot of neurotransmitter called acetylcholine. And neurotransmitter is a chemical message that's stored in these synaptic vesicles, and this specific type of neurotransmitter that your motor neurons use is called acetylcholine. And acetylcholine is a pretty basic molecule, but its purpose is to serve as a electrical or chemical signal that can bridge the electrical signal of a neuron to a muscle. Now, the infoldings of muscle cell membrane in the sarcolemma are called junctional folds that contain millions of acetylcholine receptors. And these receptors receive the acetylcholine signal as it's released from the synaptic vesicles by the axon terminal. And the neuromuscular junction consists of axon terminals, the synaptic cleft itself, and those junctional folds with the acetylcholine receptors on them. So they're looking at a picture here. We can see that filamentous extension called the axon that then branches out, and these branches form a point of communication with this muscle cell called the neuromuscular junction. If you zoom in here on the neuromuscular junction, we see that we have the expanded end of the axon here in gold contains our synaptic vesicles with acetylcholine in it. That's the little blue circles, the green circles. And when the electrical impulse travels down the axon, it gets to the end of this nerve terminal, axon terminal, and the electrical impulse or action potential here, shown in kind of this pink or purple line, it ultimately triggers the opening of these voltage-gated calcium channels. Now, a voltage-gated channel is one which opens in response to a change in charge or voltage, and these voltage-gated calcium channels open when the electrical impulse travels towards the end of the axon here. Calcium rushes into the cell down its diffusion gradient, and then what calcium does is it triggers the exocytosis of these vesicles containing acetylcholine. Now, acetylcholine gets exocytosed into this space here called the synaptic cleft, where it basically diffuses into this space. And it has the potential now to bind to receptors on the junctional folds of our muscle cell here. Remember, our muscle cell or motor end plate contains millions of these acetylcholine receptors, which can bind with acetylcholine. Now, these acetylcholine receptors are also called nicotinic receptors, and when acetylcholine binds here, it's basically a ligand-gated ion channel, which means that when the ligand or acetylcholine binds this channel, it opens up the channel and allows for these ions to flow. So, sodium is able to flow into the muscle cell, potassium flows out. However, there's much more sodium that flows into the muscle cell than potassium that leaves. So what happens is we get a lot of this positively charged sodium now rushing into our muscle cell, And this little local area of positive charge is what something called the end plate potential. An end plate potential is a little local area of depolarization or positive charge that has the potential now to maybe stimulate an action potential or electrical impulse in the muscle cell. Now, to get rid of the stimulus or to get rid of the signal here, we have another enzyme called acetylcholinesterase. And acetylcholinesterase basically breaks down acetylcholine into choline and acetic acid, and these are actually recycled by your neurons and then basically made back into acetylcholine. But when acetylcholinesterase breaks down ACH, these degraded bits can't bind with the receptor, and so these ligand-gated ion channels actually close instead of being open. So then you stop getting that current that may trigger an action potential in the muscle cell. So just to summarize those steps, step 1 was action potential goes down the nerve terminal. Step 2 is where the voltage-gated calcium channels open up. Calcium moves down its electrical chemical gradient. Step 3 was calcium entry causes exocytosis of neurotransmitter-containing vesicles. Step 4 is when acetylcholine diffuses through the synaptic cleft and binds the receptors in the motor end plate. Step 5 is when acetylcholine now causes these ligand-gated ion channels to open up, allowing for sodium to rush into the cell, causing end plate potential. And then step 6 is basically where acetylcholinesterase can break down the signal, or acetylcholine, into the degraded components like acetic acid and choline. And that way you can't, open up these channels, and so the current stops. Now, what we find then is that in the sarcolemma, we see there's a separation of charge or polarization, which means there's a voltage across the membrane. So the inside of the cell is typically more negative with respect to the outside, and the action potential is caused by changes in electrical charge distribution across the sarcolemma. So it turns out that the generation of an electrical current in the muscle cell occurs in 3 steps here. We have end plate potential, which occurs at the neuromuscular junction, and it's due to the current flowing through those ligand-gated ion channels or acetylcholine channels or receptors. And then steps 2 and 3 are parts of the action potential. We have depolarization and repolarization. Now, end plate potential is when acetylcholine is released from the motor neuron. It binds the receptors in the sarcolemma. It causes these chemically gated ion channels to open, and it allows for sodium to diffuse into the muscle cell or fiber. So some potassium diffuses outward, but not as much as sodium diffusing inward. And this influx of sodium makes the inside of the muscle cell locally, right there, more positive or less negative. That's because the charge on sodium is positive. So if you're bringing in more positive charges, you're going to make that local area more positive. And this results in a local depolarization or a local area of positive charge called end plate potential. Now, if end plate potential is big enough, it could stimulate an action potential. Now, these end plate potentials are basically a wave of sodium that come through these millions of ligand-gated ion channels at the motor end plate. And so once acetylcholine binds these channels, you get a bunch of sodium now that rushes into the muscle cell. And because of diffusion, it causes this wave of depolarization, or a wave of positively charged sodium that spreads through the inside of the muscle cell. Now, this wave of depolarization moves to the local area of sarcolemma nearby, where if it's strong enough, it may actually trigger the depolarization phase of an action potential. Now, depolarization phase of action potential is basically when there's enough end plate potential to cause these voltage-gated sodium channels to open up. Now, voltage-gated sodium channels open at a more positive voltage, and that influx of sodium through the acetylcholine channels could potentially open up these voltage-gated sodium channels, which makes the action potential unstoppable. And if this happens, then we get action potentials that spread across the sarcolemma from one voltage-gated channel to the next in adjacent areas, and it causes that area to depolarize. We call this propagation, and propagation of the action potential occurs all across the sarcolemma, down the T-tubules, and could potentially cause the opening of the release of calcium from sarcoplasmic reticulum. So looking at this diagram again, we see that this wave of depolarization, or end plate potential, spreads to a nearby patch of sarcolemma. If it's strong enough, we get this little depolarization here due to sodium. It could potentially open up these voltage-gated sodium channels that allow even more sodium to rush in. And this is the first phase of what we call an action potential or electrical stimulus. Now, this action potential spreads very fast across the sarcolemma, and it propagates by exciting other nearby voltage-gated sodium channels. Now, the next phase of the action potential, and it's actually a way for action potentials to be eliminated so you can remove the signal from the cell, is called repolarization. And repolarization is a restoration of resting conditions where voltage-gated sodium channels close, and instead that increase in positive charge within the muscle cell causes voltage-gated potassium channels to open up. Potassium actually flows out of the muscle cell down its electrochemical gradient, and the removal of positively charged potassium from the muscle cell actually makes the inside of the cell more negative, and it brings the voltage or charge back down towards a resting or negative state. Now, we call this period of time refractory, and it's basically a period where muscle cells can't be stimulated again for a specific amount of time until repolarization is complete. And ionic conditions of the resting state are restored by the sodium-potassium pumps you find all throughout the membrane of these cells. So just to look at these steps in 3 steps here, remember the first phase here is the end plate potential, which is generated due to an influx of sodium from these acetylcholine receptors at the neuromuscular junction. That wave of depolarization or end plate potential, if it's strong enough, could potentially cause these voltage-gated sodium channels to open up, allowing an influx of sodium, which is the depolarization phase of the action potential. These action potentials spread really rapidly, and there's 2 phases of the action potential. Depolarization, which is due to the opening of these voltage-gated sodium channels, and then repolarization, which is the opening of these voltage-gated potassium channels, which allow for potassium to leave the muscle cell, thereby making the inside of the cell, again, more negative, back towards a resting state. So this is showing the voltage trace of an action potential. Like if you can record the voltage changes or electrical changes of a muscle cell over time, you'd find that resting voltage is around negative 90 millivolts, and that once an action potential occurs, these voltage-gated sodium channels open up, sodium rushes into the cell, makes the inside of the cell more positive due to an entry of positively charged ions here. And then near the peak of the action potential, voltage-gated sodium channels close, and voltage-gated potassium channels now open. And that due to an efflux or exit of potassium, we see that it makes the voltage inside of our muscle cells more negative because now you're removing positive charges. And that efflux of potassium brings the voltage back down towards a resting state. And this little blip in voltage is the action potential. Now remember, these actually spread very quickly across the muscle cell, and it's these little blips in voltage or action potentials that actually trigger sarcoplasmic reticulum to release calcium. So we call this excitation-contraction coupling. And it's the events that transmit action potentials along the sarcolemma, that's the excitation phase, coupled to the sliding of myofilaments in the sarcomere, which is the contraction phase. Now, the action potentials propagated or spread along the sarcolemma down the T-tubules, where voltage-sensitive proteins in the tubules stimulate calcium release from the sarcoplasmic reticulum. That calcium eventually leads to a contraction because it can actually bind with troponin, move tropomyosin out of the way, and allow for the myosin heads to bind to the actin active binding sites. Now, action potentials are brief. They're about a millisecond, so 1/1000th of a second, and they end before the contraction is even seen. But they spread rapidly and eventually could lead to a contraction. So just to summarize those steps, remember the action potential gets to the nerve terminal, triggers the exocytosis of neurotransmitter-containing vesicles, acetylcholine binds to nicotinic receptors, causing an end plate potential that spreads to the nearby patch of sarcolemma. If an action potential is generated, it spreads down across the sarcolemma, down the T-tubules, and that causes the release of calcium from the sarcoplasmic reticulum now in the sarcoplasm, and that calcium can bathe the myofibrils now, where calcium can go on over, bind with troponin on the thin filament. Troponin pulls on tropomyosin in the thin filament and removes the inhibition from those actin binding sites. That way the myosin head can actually bind with actin. It actually forms a cross bridge, and it pivots to basically pull on the thin filament in a direction towards the M line on the sarcomere. So at low intracellular calcium, tropomyosin blocks the active sites on actin. And the myosin heads can't attach, therefore you can't get a muscle contraction, so the muscle fiber or cell stays relaxed. Voltage-sensitive proteins in the T-tubules change shape and causes SR to release calcium into the cytosol. But at higher intracellular calcium concentrations, calcium binds to troponin. Troponin changes shape, allowing tropomyosin to move out of the way so that myosin can bind with actin, forming a cross bridge, and cycling is initiated, where basically the myosin heads cycle really rapidly by moving around and basically using the chemical energy in ATP to produce a muscle contraction. And when nervous system stimulation ceases, calcium is pumped back into the sarcoplasmic reticulum, thereby ending the contraction. So to summarize these steps, remember the action potential travels down the T-tubules, triggers the opening of these voltage-gated calcium channels, which allow for calcium to spread from the sarcoplasmic reticulum down towards the sarcomere, where it binds with troponin. Troponin pulls on tropomyosin, moves tropomyosin out of the way, allows for the myosin heads to bind to form a cross bridge. Now, the 4 steps of a cross bridge cycle are, first of all, the cross bridge formation, which is a high-energy myosin head bond with actin thin filament. Now, the working or power stroke is when that myosin head pivots and pulls on the thin filament towards the M line. And then you get cross-bridge detachment when a new ATP binds with the myosin head. Myosin heads detach and then recock, basically through the hydrolysis of ATP into a high-energy state, which could potentially be used in the next cross-bridge cycle. What we find is actually this is a cycle where, as long as calcium's bind to troponin, tropomyosin's out of the way, and your active sites are now exposed, the myosin head can form an attachment. It actually pivots, kicking off ADP and inorganic phosphate through that power stroke or contraction. And then when a new ATP molecule binds with the myosin head, that myosin head can unbind and then use the chemical energy in ATP to recock back into a high energy state, where as long as the active sites are exposed, again, myosin head can bind and the whole thing recycles and repeats. Now, one thing that's important to note is you need ATP for this process to continue, and you especially need ATP to get the myosin head to unbind. So one thing that's interesting here is that in rigor mortis, or stiffness after death, we see that due to a depletion of ATP, the myosin heads can't unbind from actin. Instead, they stay in the bound or cross-bridge state, which makes muscles more stiff. This is actually what's going to explain the stiffness of muscles after death.